Hello dear friends, welcome to Toppscholars, the Smart Learning Gap. Today we will start with a very interesting topic which is gonna help you in your upcoming examinations. So the topic is interphase of cell cycle. You just take a pause and take a screenshot of this slide because we are gonna deal with all of these things in our lecture. Before we deal into all of these things, we need to understand what is cell cycle. So herein, what do you mean by the term cycle? Basically, you have seen the bicycle, right? But here we are not referring to that. In the bicycle, you see there is wheel, right? When you ride a bicycle, it keeps on moving. The wheels are moving, right? In a cyclical pattern. So here in cell cycle, we mean that our cell is going in some cyclical events. Okay, what are these events? Let us have a look. So over here, as you can see, this shape is of a circle, right? And these are the respective phases in which a healthy, normal cell will continue its life cycle, right? So over here, let's start with this G1. You need to remember these terms. These are the acronyms for actually the bigger words. So here, just keep in mind that we have G1. Then this arrow shows that after G1 is completed, our cell will enter into S phase, okay? And after this, we will have G2 phase, after which we will have the M phase, okay? And then some cells might go into another phase which is called as G0, and they can also be called back to this continuous cycle, that is, they will enter G1 phase. Okay, so basically the thing to remember over here is the sequence. Okay, we will always keep the sequence as G1, then we will have S, then we will have G2, and then we will have M. Okay, after M, the cell might or might not go into the G0 phase. Now students, you have understood the sequence. We have G1, S, G2 and M. Let us see what are the events undergoing in these phases. Okay. First of all, you need to understand that this G letter is used for the word gap. Okay. So basically, we have G1 phase and then we have G2 phase, right? In between, we have S phase. Over here, the letter S stands for the word synthesis. Now, what is being synthesized? Yes, it is the DNA. And what is DNA? DNA is the genetic material of most of the organisms, right? Only we need to exclude some RNA viruses. Otherwise, all the cells have DNA as the genetic material. And for a cell to divide, for a cell to multiply into more cells, we need to have DNA in it. Why? Yes, because it is genetic material. It is having all the informations to run the cell and to run the organism itself, right? So here we have G1 phase, then we have S phase, then we have G2 phase. Now there are certain events that are undergone in G1, S and G2 phase. Now remember, we have started our topic of interphase. For that, you need to understand this M phase, okay? Herein, depending upon the cell type, we may have M standing for mitosis or M standing for meiosis phase. Okay. In this diagram, we are given that M phase is nothing but mitosis. So, between two consecutive M phase, you will have a phase, okay, which is called as interphase. Since it is in between two consecutive M phase, hence we are giving the name interphase. Okay, and it comprises of consecutive phases of G1, S and G2. Okay, now one thing to remember over here is this term, which is cytokinesis. Okay, herein this cyto part means cytoplasm and kinesis is referring to its division. So, if we are going from one cell to two cells, then obviously we need to divide the cytoplasm as well, right? The cytoplasm amount present in one cell will be distributed into two daughter cells. Hence, we have this phase of cytokinesis just after we have divided our cells in M phase. Okay. So over here we have just learned that what are these letters standing for. G is standing for gap. Here we have in interphase gap 1. Then S phase, S is standing for synthesis. What is being synthesized? It is DNA. And then we have G2 phase wherein it is gap 2. Understood? After this we will have the M phase, which might be mitosis or meiosis depending upon the cell type and need. Okay. After that there is a short period which is of cytokinesis where we will divide the cytoplasm. Understood? Now remember we have started our topic with interphase. Okay. This is the topic that we are concerned with today. Over here, do you know that biology is a general term, right? In here we have different disciplines. For example, we have cell biology and then we have molecular biology. Okay. Now we just said that between two consecutive M phases, there will be the presence of interphase. So what are the things going on in it? Is this interphase really significant or not? What are we actually doing in that interphase part of the cell cycle? That all comes into mind, right? Now over here, we are stating that a cell biologist, that means a person who is learning or studying the cell biology. Those persons will refer to activity in terms of cell division. That means in the cell cycle, cell biologists will say that activity is going on in M phase only. Why so? Because M phase has cell division and they are referring activity to cell division only, right? We have another one which is molecular biologist. So this person, molecular biologist, will refer to the events that are happening inside the cell as activity. Okay, he is not concerned with whether the cell divides or not. He or she is just concerned with whether there is activity going on inside the cell or not. So basically, a molecular biologist might not say that M phase is active phase. Okay, we will see all of those things in just few moments. This is just to clarify you the meaning of activity with respect to cell biologist and with respect to a molecular biologist. Okay, now let us go ahead. Over here you can see these are the phases which we will learn in just few moments. Just wait a minute. Over here this is the cell cycle which shows you the sequence of events in a cell. So over here this part is the M phase. So over here in this cell cycle we have this part which is referred to as mitosis and this part is interphase. So here this is the pie chart for happy and healthy cells. Remember, healthy cells which are in control, which are in regulation will undergo the cyclical events which are present in the cell cycle. So for defining a cell cycle, you can say that it is a sequence of events which are undergone in a healthy cell. Okay. So over here, this is the pie chart. We can see this cell cycle as a pie chart in which a very small proportion is for M phase. Okay. Remember, M can be mitosis or it can be meiosis. Then we have this rest of the part which is the interphase. Okay. So as we just said, this part is interphase and this small proportion is of mitosis or meiosis, that is M phase. So here we have our interphase. Let us have it in a diagram. Okay. Over here you can see that first of all we will have G1 phase, that is gap 1. Then we will have S phase, that is DNA synthesis. And then finally we have G2, that is gap phase. So finally we can say that we have G1 which is standing for gap phase 1. Then we have S phase which is standing for synthesis. And then we have G2 phase which is standing for gap 2 phase. And over here we have the M phase. In this part we will have the cell division going on. And this whole part of this blue color is comprising your interphase. Okay. Now over here S phase we have the synthesis of DNA. So over here as we just saw we have the mitosis or meiotic phase in which the cells will undergo division. So over here students we have the M phase. You can see this part is referred to as mitosis in this figure. This part is, as we can just imagine, or it is self-understood that this part is interphase. Just see at these pictures which is drawn. You can see this cell is smaller in size, right? Which then increases in the size. Then what are they doing? They are weaving, like we weave the wools, right? They are weaving the DNA. That means DNA is being synthesized, right? And then they are all set with a ready backpack. That means they are ready for their next phase, which is mitosis in this case. Okay. So here we can say that in interphase, the cells are getting ready for doing the cell division. Okay. As you can see over here, we have one cell. Okay. That is undergoing some events, and then magically we have two cells present over here. Right. And these two cells are yet connected, which will be divided after following the phase of cytokinesis. Remember? So, after division, we have two different daughter cells which are separated. Okay. And these are coming from one parent cell. Okay. So, basically, this was the interphase and this is the mitosis. You can take an analogy of you getting ready for your schools or your colleges. Okay. You get up in the morning and then you start tidying up, right? You take a bath and then you will get dressed, you will have the breakfast, you will fill in your backpack with books or registers or textbooks which are needed in your school, right? For the lectures, obviously. So, the whole process in which you are getting ready to go to school will be the interphase, and you actually going to school and attending the lecture will be the mitosis. So, this is just an analogy for you to understand what is actually happening in the cell cycle in these two major phases, that is interphase and M phase. Okay. So, students, just now we have said about cell biologist and a molecular biologist. Over here, interphase is actually a resting phase as per the cell biologist. Can you guess why? Yes, because a cell biologist was referring activity to cell division, and in interphase, the cell is just getting ready. It is not dividing. Hence, a cell biologist will say that interphase is a resting phase. It is a calm phase in which no activity is seen. But this is as per the cell biologist. What will a molecular biologist say? A molecular biologist will say that this interphase is a very active process. And can you guess why is this so? Yes, we have just talked, right? A molecular biologist is referring activity to the events that are undergone in the cell. And we just saw that a cell undergoes a lot of events. That is, it gets ready for the upcoming cell division. That means it is an active phase as per the molecular biologist. Okay. So, please remember these two terms. Activity is cell division for cell biologist and activity is events inside the cell happening for a molecular biologist. Okay. Now, let us go ahead. Over here, you can see this is the cell which is present in interphase. Rather, this one is an image from your G1 phase. Okay. How we can infer that? We will see in just few moments. Okay. So, over here, let us have the interphase definition finally. Interphase is the phase where the cells grow in size. As we just saw those images, right? That emoji was very small in the beginning, but after some time, the next emoji was larger in size, right? That means that cells are growing in size. Then it is duplicating its content like the DNA, okay? As we have already studied about S phase, in that DNA is being synthesized, right? That means DNA duplication or DNA replication is going on in interphase itself. It is not undergone in M phase, okay? Then along with DNA, organelles are also duplicated. Now, what are these organelles? Yes, similar to like we have organs in our body, cells have organelles in the cellular structures, okay? What are these organelles doing? They are performing various functions to carry out all the cellular activities. Can you give me some examples of cell organelles? Yes, mitochondria, ribosomes, we have Golgi apparatus and many more, right? All of these are organelles. Now just imagine if I have one cell, okay, it is having the required number of organelles for one cell only. But following division, there will be two cells, right? And the organelles must be duplicated, they must be doubled so that they will be evenly distributed in the two daughter cells, right? That's why in interphase we have duplicated the DNA, which is to be separated and segregated into two daughter cells, right? Similarly, we have also duplicated the organelles, which is also essential for the survival and functioning of the daughter cells, right? So basically we can say that in interphase, the cell basically prepares itself for the upcoming division phase, okay? Hence, we were just saying that interphase is in between two consecutive M phase, okay, as the name suggests. And we have seen the sequence as well. We have G1, then S, then G2 phase, which will comprise the whole of interphase. In between, we have G0 phase, which we will just see in some few moments. Okay, going ahead, you can see we are now dealing with the separate phases which are comprising the whole of interphase. The very first thing is G1 phase. Now, this is gap 1. What do you mean by gap 1? Yes, it is the first gap which is taken by the cells after division. Okay, so G1 phase is just after the M phase and it precedes the subsequent S phase. Just wait a minute, over here we will have the diagram as well. So over here you can see this one is the first gap, that is G1 phase. After which it is coming after mitotic phase, right? In mitotic phase, we need to remember this acronym which is PMAT. Okay. Now, what are these things? What are these letters standing for? We will be learning in our future videos which will deal with mitotic and meiotic phase separately. Over here, we are concerned with the interphase now. So, this first gap phase is just after mitotic phase and after this we have the S phase. Okay, remember this sequence as well because in your examinations, sometimes only this sequence can also be asked. Okay, so what are the events that are undergone in gap 1 or G1? Let us see. The first gap period or the first growth period is the G1 phase, as we have just said. It starts immediately after the cell division phase, that is M phase. Synthesis of RNA, that is mRNA, rRNA and tRNA is undergone in this G1 phase, and also membranes is being synthesized. Now, do you know what is the usage of RNA in our cells? Yes, if you remember the central dogma, okay, we have DNA from which we will be preparing our RNA, right? From which we will have our proteins, right? So, from this DNA, we need RNA using which we will produce the proteins, and proteins are necessary for cellular functions, right? Hence, we require these three different kinds of RNAs. M stands for messenger RNA, R stands for ribosomal RNA. This is the constituent of ribosomes, which are the protein making machineries, right? And this tRNA, here T stands for transfer RNA. Okay, so different kinds of RNA is being synthesized and membrane is being synthesized in your G1 phase. Over here it is written that cell will duplicate its DNA. Where do you think this thing will happen? Yes, it will happen in S phase. Hence there is a arrow mark given. Okay, at this phase we will have DNA duplication. Okay, let us see the S phase now. Over here you can see these are the DNA. There are respective base pairs seen, right? In S phase, the DNA is being duplicated. DNA is synthesized or replicated. And histone proteins are also synthesized. That means DNA along with histones are being synthesized in S phase only. Now, I have a question for you. What is this histones? Yes, let us see what are these histones basically. Do you remember that for any interaction, we need to have opposite charges, right? If we have positive and positive, it will repel. And if we have negative and negative, it will also repel. So, for DNA to be accommodated inside a cell, DNA is very huge, right? If you remember, if you extend the DNA which is present inside one cell only, it will measure up to 2 meters. And our cells are very small. Okay, so we need to accommodate that much amount of DNA inside a very small cell. For doing that, we need to condense our DNA structure. And for that, we have the use of histone proteins. Okay, over here you can see DNA is approximately 2 meters and our cell size is in micrometers. That means in the order of 10 raised to minus 6. Okay, 10 raised to minus 6 meters it is. And over here DNA is of 2 meters. So how will it adjust? It will adjust because of histone proteins. Along with other models, one is of histone interaction. So DNA is called to accommodate itself in the nucleus of the cell. What will happen? Basically, DNA is negatively charged. Why is DNA negatively charged? Yes, because it has phosphate groups which impart the negative charge to the DNA molecule. So, DNA is having negative charge and these histones for attraction to that DNA will need to have positive charge, right? Since these are proteins, these have positively charged amino acids present in them which imparts a net positive charge to histones. And now as you can imagine, we have positive charge for histones and negative charge for DNA. Now they can wrap onto each other and form a condensed structure. Okay, so here we can say that histones are positively charged and therefore electrostatic interactions will occur between DNA and histone which will lead to the winding of DNA on histone proteins. You can just imagine a thread roll, okay? The base is your histone proteins and the thread that you are winding is actually the DNA, okay? So how much compact structure is that of a thread roll, right? Similarly, the DNA is wrapped around the histone proteins in order for it to accommodate inside the nucleus of the cells. Okay. So, now let us go for G2 phase. So, what have we learned so far? We have learned that in G1 phase, we will have RNA synthesis, we will have membrane synthesis, and then we have learned about the S phase in which there will be duplication of DNA and histone proteins are also duplicated in S phase only. Now we have completed our G1 and S phase. We are moving for G2 phase. Okay. So this one is gap 2 phase. This is coming after the S phase, that is synthesis phase, and it is just before the division phase, that is M phase. So what you think the cell will be doing in G2 phase? Yes, it will get ready for the upcoming division. It is obvious. So what will it be doing? Let us see. As we just saw that G2 phase is the second growth phase and in here the nucleus increases in volume. Okay, so if the nucleus is this much, let's say for analogy, it will just increase in its size so that the DNA will be accommodated properly and the cell will get ready for the upcoming cell division phase. Okay, then over here the cell undergoes metabolic activities which are essential for cell division. Okay, for cell division, the cell will require a lot of energy, a lot of proteins. Those all things will be done in the G2 or gap 2 phase. Okay, this is the second growth phase or the second gap phase, right? Going ahead, you can see this one is the G1 phase which we have already studied. Then we have S phase wherein chromosome duplication or DNA synthesis will occur. Okay, so over here, what is the term chromosome? Yes, it is the highly condensed and compact form of DNA. Okay, then finally we have the G2 phase, after which we will have the cell division, that is M phase. Over here you can see we have two things, one is cyclin and another one is CDK. What is this CDK standing for? Yes, it is cyclin dependent kinase. Okay, so proper flow of all these events in a cell, some proteins are there which will keep the regulation in proper manner. Okay, these are cyclins along with other proteins and these are proteins which are cyclin dependent kinases. As the name suggests, they will have kinase activity, meaning they will transfer phosphate groups to different targets. Okay, but they are dependent on cyclins for their function and all of these things is happening throughout the cell cycle. Okay, So over here we have learned that in G2 phase, metabolic activities essential for cell division will occur and also the nucleus will increase in its size. Okay. Going ahead, proteins necessary for cell division will also be synthesized. Then if there is more amount of RNA required, then that will also be synthesized. Okay. Now in animal cells, a daughter pair of centrioles will appear near the pre-existing pair. Centrioles appear on both the poles of the cells just before the division. Okay. Now say for example, we have one pair present on one pole and one pair present on another pole. Now this is the case for one cell, but after the division we will require two pairs of centrioles at each pole, right? So that it can be separated properly in the two daughter cells. Hence, centrioles also appear near the pre-existing pair in animal cells. Plant cells don't have centrioles. Please remember this key aspect or key distinguishing point between animal and plant cells. Okay. Going ahead, let us discuss about M phase briefly. Okay, we are just introducing you to the concept. We will deal with more details in our upcoming videos. So over here, M phase can be mitosis or meiosis depending upon the cell type. Meiosis is undergone in, yes, in sex cells. Why? To produce the gametes. Okay, this meiosis is also referred to as reductional division. These things you already know, but you will learn in more details in this 11th standard topic. Mitosis is also known as equational division. Okay. So in mitosis as well as in meiosis, you will have replication phase, after which there will be cell division. Okay, as you can see. But in mitosis, after cell division, we have two daughter cells produced, which are having same DNA content as the parent cell. But in meiosis, we have four daughter cells produced from one cell. Okay, so here the DNA content is halved. Hence, we said that this one is reductional division and this one is equational division. Okay, going ahead. So over here, let us have a beautiful animation for the M phase. In here, you can see these are the cells which are showing the process of meiosis. So as you can see over here, from one parent cell, it is forming four daughter cells. Okay, say for example, if I have 2n chromosomes in the parent cell, then after following meiotic division, in each of these four cells, I will have only n amount of chromosome. Okay, hence we say that this meiosis is a reductional division. In meiosis, we have two different phases. Okay, first is meiosis 1 and then we have meiosis 2. We will see all these in future upcoming videos. For here, let us now see the mitosis. So in here, as you can see, we have one parent cell and from that we are forming two daughter cells. In here, if I talk about the chromosome scene, okay, chromosome is 2n in parent cell and in daughter cells also we have 2n chromosomes present. Hence, mitosis is an equational division. Understood? Now, as we just said, M phase is equational division. In M phase, as we just saw, we have one parent cell which will be divided into two equal daughter cells. Okay? Hence, we are referring to it as an equational division. Okay? Over here, you can also see we have two cells which are being produced from one cells following mitotic division. Okay? Now, going ahead, we have a detailed description for your mitotic phase. We just saw that we have an acronym which is PMAT. Okay. Over here, P is standing for prophase, M is for metaphase, A is for anaphase and T is for telophase. Okay. Over here in prophase, you can see that the DNA is being compacted. Okay. Over here in interphase, chromatin is present, which is a loosely packed DNA structure. Okay. But in prophase, we are going for a compact DNA structure. Then in metaphase, this meta is for middle or equatorial plane, right? Over here, the DNA is being lined up in the equatorial line, which is meta plate. Then in anaphase, this chromosome chromatids are being pulled towards the pole. That is, they are pulled away from the center. That A is for this anaphase. And finally, in telophase, you can see there is reformation of the nuclear membrane and the cell is ready for cytokinesis, right? So this is all the events which are undergone in mitosis. We will see all this in our separate video lecture as we have just said. Then we have meiosis, which is the reductional division. Why so? This is reductional in terms of the chromosome number. Okay, if the parent cell is having 2n, then the daughter cells will have n. That means if the parent cell is diploid, then the daughter cells will be haploid. Okay. Over here, you can see there is meiosis 1 and then we have meiosis 2. In meiosis 1, there is prophase 1, there is formation of tetrad, and then finally we have reductional division, after which we will have meiosis 2. Remember, mitosis is equational division and meiosis 2 is also an equational division. Okay, and meiosis 1 is actually a reductional division. We will see all these further, but over here you can just take a note that mitosis and meiosis 2 are same and meiosis 1 is actually the reductional division, for which we are referring to meiosis as whole, a reductional division. Okay. So going ahead, let us see the G0 phase. We have seen in our cell cycle, right? Some cells might go or might not go in G0 phase. What is this G0 phase? This is a quiescent phase. What do we mean by quiescent phase? So basically, some differentiated cells like, say for example, the neurons or the heart cells, that is cardiomyocytes, okay, these cells have lost their ability to divide. That means they are no longer usually undergoing the process of cell division. So they need not be in the cell cycle, right? Because if they are present in G1, say for example, they will have to go for S phase, then for G2 phase, then for M phase, right? This holds true for any position in the cell cycle. But these cells don't even want to divide. Henceforth, they are present in G0 or quiescent phase, which is different than the whole cell cycle phases, and it is referred to as quiescent phase, okay, because the cells which are present in here will not undergo cell cycle usually. But if they want to, in some stimulus or in some conditions, they might re-enter the cell cycle. They can, okay, but usually cell division is not occurring for these differentiated cells, right? So this was all about the G0 or quiescent phase. Some people also refer to it as inactive phase, but mind it, it is not an inactive phase as such, because your neurons are actually doing a lot of work, right? Your heart is also doing a lot of work. So how can we say that in G0 phase the cells are inactive? No, they are active. It's just that they are not dividing normally, okay? They are not going through the cyclical events which are present in your cell cycle. Going ahead, there are also some cells which don't have the gap phases itself. That means G1 and G2 phases are absent. So what do you think? Which cells will have absence of gap phases? Yes, those cells which need to divide very rapidly. Do you remember that zygote is formed after the fusion of male and female gamete? Yes. What happens after that? The zygote repeatedly divides to increase its number. It will go from 1 to 2, from 2 to 4, then 8, 16. It will form a 32-celled stage and so on and so forth, right? Divisions are very rapid in a zygote. To do what? Yes, to form the embryo, right? So there are some cells which don't have gap phase itself, okay? For example, we have zygote. What is happening in it? It has alternating S and M phase without the gap phase. In S phase, what will happen? Yes, the DNA will replicate, and in M phase, the cell will divide. So basically, replication, division. DNA replication, cell division. DNA replication, cell division. This is what keeps on happening in zygote, okay? So over here, you can say that if this is the pie chart for our cell cycle, then half of the part is for S phase and half of the part is for M phase. There is alternating M and S phase going on, okay? So we have seen normal cell cycle, right? We have seen what is interphase, what are the events in interphase, that is G1 phase, S phase, G2 phase, right? Then we have seen that there are some cells which don't undergo cell cycle regularly. Instead, they are being differentiated for their specific function. Say for example, the neurons and the heart cells. So these are present in G0 phase. But then we have also studied that there are some cells which don't have the gap phase itself. Okay. These cells are rapidly undergoing division, and in this case we have learned the example of zygote. Okay. Now let us see what is the amount of time which is spent in each phase of the cell cycle. As you can see over here, we have the G1 phase, which is gap 1, just following the M phase. Okay. This is the largest phase which is present in cell cycle. It extends up to 6 to 12 hours. Okay. And over here we can say that it is taking up to 8 hours or even more. Okay. So remember, G1 is the longest phase in the cell cycle. And which is the shortest? Yes, it is the M phase, as you can see over here. M phase is the shortest phase in the cell cycle. A cell spends minimum amount of time in division. It spends 95% of time in interphase only. And in that also, most of the time is spent in G1 phase. Okay. In S phase, we have 6 to 8 hours, and in G2 phase, we have 3 to 4 hours. So this is the amount of time which is spent in each phase. Now over here you can see we have DNA is equal to 2n. Here we have DNA being transformed to 4n from 2n. In G2 we have 4n transforming into 2n. Now remember students, let us see over here. This is G1 phase, this is S phase, this is G2 phase, and here we have M phase. Okay. Say for example the DNA content. Okay. The DNA content I am referring to as 2C. And the chromosome number I am referring to as 2n. Okay. So this is diploid condition, and this is the amount of DNA that is present in the cell. In S phase, what will happen is our 2C will become 4C, which means in S phase we are duplicating or replicating the DNA. This thing is very important for you to understand what is going on in interphase and in M phase. Okay. So in S phase we have duplicated the DNA content, but the number of chromosome remains same. Okay. So here we have 2n only. Over here you can see we have written 4n. Here n is representing the amount of DNA, not the chromosome number. Some people refer to small n as the amount of DNA. Some people refer to small n as the number of chromosomes. You need not get confused. Okay. Hence, I am using different letters only. Just remember, we are using C for the amount of DNA and N for the number of chromosomes. Okay. So, in S phase, the amount of DNA is doubled, but the chromosome number remains the same, that is 2n. So, what will happen basically is that if I have this much DNA present in G1, what will happen in S phase is I will have double the amount of DNA. Okay. But the chromosome number is same. How is this so? Students, chromosome number is always counted as per the number of centrioles. Okay. So over here if I have one centriole, then I will count it as one chromosome. Over here if I have one centriole, this will also be one chromosome. Okay. But I have doubled my DNA content. This is the trick which you need to understand. No one will ask you separately, but you need to better understanding the mitotic and meiotic divisions. Okay. So over here in S phase, the DNA content is being doubled, but the chromosome number remains the same. Okay. Then in G2 phase, again we will have 4C only. This will be 2N only. And if the M phase is mitosis, okay, then I will have 2C in different 2 cells, right? And over here I have 2n because a diploid cell forms 2 diploid cell in mitosis, right? Let us write mitosis over here. So this one is mitosis. Then let us see what will happen in meiosis. Let us denote it with M itself. So what will happen in meiotic division? This 2n with what we have formed in mitosis will not be formed in meiosis. Here we will have n. Right? Remember we have 4 haploid cells which are being formed. Okay. And the DNA content will be C. Right? So, in 2 cells NN and we have CC, and in other two cells we will have NN and CC. Okay, so you can see that we have formed 4C, right? From 4C we have came to C, and from 2N we have came to N in meiotic division. For mitosis, we have came from 4C to 2C, and we have came from 2N to 2N only. What is this C? This is the amount of DNA present. What is this N? N is the number of chromosomes present. Okay. Please try to remember these all things as per the amount of DNA and as per the number of chromosomes as well. Okay. So over here it is written that mitosis lasts for 1 to 3 hours, which we have already studied, right? The shortest time spent is in M phase. Now let us go for some previous year questions from this topic. This topic is very important. Now we have just included questions from your interphase. There are many questions coming from mitosis and meiosis as well. So you cannot opt out this topic for your examinations. Okay. So the very first question is of matchup, and this came in this year only in 2023. Okay. Let us see. So we have list 1 and then we have list 2. We just need to match them properly, okay, appropriately. That is the trick, right? So in first we have M phase, then we have G2 phase, third one is quiescent stage, and fourth one is G1 phase. Now this quiescent stage is nothing but the G0 phase, right? Let us have the entries in list 2. First one is proteins are synthesized, okay. Second one is inactive phase. Third one is interval between mitosis and initiation of DNA replication, and fourth one is equational division. Now please just forget everything and tell me, what is this equational division? Yes, it is mitosis. We have said it many times in our lecture today. So M should be matched with 4, right? Mitosis is the equational division. Now let us go for another simple thing, inactive phase. And over here you have quiescent stage. Yes, some people were saying, right, that G0 is an inactive phase with respect to cell division, not with respect to activities, right? So C should be matched with 2 only, right? So we have 2 matchups already done. Now let us go for this one. This is more important and that is more easy as well. You just need to remember the events in the cell cycle. So this one is interval between mitosis and initiation of DNA replication. That means this phase is in between M, and initiation of DNA replication occurs in S phase. Okay. So which phase is in between M and S? Yes, it is G1. So D should be matched with 3, right? What was the sequence, friends? It was G1, then we have S, then we have G2, then we have M, and this cycle will again be repeated in normal healthy cells which are undergoing cell division. So the interval between mitosis and initiation of DNA replication becomes G1. Now what you are left with? You are left with G2, and in here proteins are being synthesized for the coming cell division phase, right? So the sequence we are looking for in our correct answer will be 4, 1, 2, 3. Okay. So friends, we are looking for the sequence 4, 1, 2, 3, right? Let us see where it is present. Here we have 2, so this one is incorrect. Second option we have 3, so this one is also incorrect. In third option we have A matched with 4, so this one is right. And in fourth option we also have A matched with 4. So just by looking at the answer for A, you can eliminate 2 options. And for claiming the correct matching for B1, you can select your correct answer and you can easily grab your 4 marks. Okay. Our sequence was 4, 1, 2, 3. Hence we will go with the fourth option as the correct answer for this question. So over here, let us highlight it. This one is the correct answer. So as you can see, this question was very easy. Just by knowing the interphase and the events, you can easily grab these marks. Okay. Next, let us see one more question. So here also there is matchup, and this has come in year 2021. Okay. So here also we have list 1 and list 2. First entry is of S phase. S stands for synthesis. What is being synthesized? DNA, right? So something related to DNA should be looked upon in the list 2. Let us go for B, that is G2 phase. Okay, gap 2, proteins will be synthesized, which will be required in cell division, right? Third one is quiescent stage. This is G0. You should know all these things at the tip of your tongue. Okay, that is very simple. So you need to just revise 1 or 2 times, keep doing the questions, keep doing your model papers, and you will be very set for your upcoming examination. Let's go for fourth entry, which is G1 phase. This is the gap 1 phase, right? Let us go for the entries in list 2. Over here, proteins are being synthesized. So yeah, we will go for G2 phase, right? We have just seen. Second entry is for inactive phase. Yeah, this is G0. We have just seen, right? Third entry is for interval between mitosis and initiation of DNA replication. Again, this thing is repeated only. So, this one is for G1 phase. Right. Last entry is for DNA replication and we have S phase for that. So, A should be matched with 4. B should be matched with 1. Again, what is the sequence that we are looking for? Yes, it is 4, 1, 2, 3. Okay. This sequence is present over here. 4, 1, 2, 3 is given in third option, right? So, the correct answer becomes your third option. As you can see, the question was repeated, right? The same things came in 2 years, right? If, say for example, you have just appeared for 2021 paper and you have encountered this question, then it will be very much easier for you to solve the 2023 question, right? So, basically, if you practice more questions, there is high chances that questions similar to those things will come in your actual examination, and that's why we have many practice questions on our application for you to solve and learn and practice. Okay, and these questions might actually come in your upcoming examination in some or the other form. Basically, you will be get trained for those things. So friends, I hope you have understood the details of the concept that we have talked in this session. Thank you for your attention.